everyone, so this is our video on our field trip at Taman Wetland Putrajaya. So this is us hearing a briefing from the facilitator. Then after having the briefing, we have an explorers held by the facilitators of Taman Wetland Putrajaya for us to gain more knowledge about variants of species of plants in Taman Wetland Putrajaya. So now we are looking for the species stated in the explorers for us to win the explorers. For your information, this plant is not just a random plant, but it is being planted to catch all the trashes that are being dumped into the lake. Then we found the Phoenicopterus chilensis, also known as baby flamingo there. Next, we went to Taman Botani Putrajaya. The tour guide said that there are 7 species of fern there. Then he showed us air mata pengantin, which is a rare plant. Uniquely, we also found deers there.
Moving on to practical 1, basic concepts of statistic and variation. The objective for this practical are to calculate and describe the following descriptive statistics, to understand the concept of correlation and to determine the relationship between the two variables. In this practical, we need to take 10 leaves to measure and record their length and width. Then we prefer 2 sets of leaves. We also calculate the mean, variation, and standard deviation. So here are the measurements for all 3 sets of leaves. Here are for set 1, set 2, set 3. Here is the graph for this practical, the length versus the width of leaf. And based on this graph, there are no strong relationship because of the natural variation, different species, and environmental factors that affect the leaf shape. Errors in measurement also can cause inconsistency. So next, we will move on to the post-lab questions.
Set 3 has the highest mean width, which is 4.5 cm, followed by Set 2, which is 4.32 cm, and Set 1, 3.83 cm. The width of Set 2 and 3 are relatively close, a difference of only 0.18 cm, while Set 1 has a noticeably smaller mean width. Although Set 1 and 2 have similar means in length, Set 2 has a higher standard deviation compared to Set 1, suggesting that leaf length in Set 2 vary more widely around the average. So this indicates that even though Set 3 has the shortest leaves, they are wider than those in Sets 1 and Set 2. This may suggest a different leaf morphology or adaptation. For example, Set 3 could be a shade plant that needs broader leaves to absorb more light.
So guys, what is variance actually? Variance is a statistical measure of how much values such as weight or length differ from average, which is mean. So, what's the relation between leaf width and length with variance? High variance means there are many leaves that are in different sizes. Meanwhile, for low variance means that there are many leaves that are in same sizes. First, to understand about the natural variation. Second, it is for accurate statistics. And lastly, is to detect pattern and correlation of the population.
Our study site was Taman Botani, where we work in the middle of the forest for both sites. The main objective was to introduce the quadrant method, enhance our observation skills, and understand how biotic and abiotic factors influence the plant distribution. We used basic tools such as rafiaring, measuring tape, and set up 2 10 by 10 meters quadrats, which is a modification from 5x5 size. In Quadrant 1, we recorded species such as Roystona regia, also known as the royal palm. This species is typically found in tropical forests and is known for its tall trunk and adaptation to well-drained soils. We also found Dryobalanopsis sumatranus, a dipterocarp species valued for its hardwood and role in maintaining canopy structure. In Quadrant 2, species like Hopia odorata appeared. This tree is known for its resistance to flood and is often found near riverbanks, showing how abiotic factors like soil moisture can influence where species grow. In other species, Neobalanacarpus haimi, also known as chengal, is important ecologically and economically due to its durable timber and role in supporting biodiversity. In Quadrant 2, species like Hopia odorata appeared. This tree is known for its resistance to flood and is often found near riverbanks, showing how abiotic factors like soil moisture can influence where species grow. In other species, Neobalanacarpus haimi, also known as chengal, is important ecologically and economically due to its durable timber and role in supporting biodiversity. By comparing both quadrats, we noticed that species like Pomatia pinnata were present in both, while others were unique to each site, showing variation in species distribution due to microclimate or soil type difference. In conclusion, this practical helped us better understand the structure of plant communities, the importance of biodiversity, and how ecological factors drive plant distribution and abundance in a forest ecosystems.
This practice, a quadrat is a marked square area. We use 10 meters by 10 meters that is for unit sampling to study the distribution and abundance of the plant species in these forest types. So it allows us for systematic observations and recording of the number of individual plants and their species and other relevant data. So quadrats are used here to estimate the population, size, density, and species diversity that will help for us to recognize the biotic and abiotic components in this community. What are the two requirements for quadrat? First is random and representative quadrat. It is to avoid sampling bias so that it requires all the habitat. Second is standardized size and shape of quadrat. Why do we need to standardize the shape of quadrat? Because we need to have consistent data collection. And disadvantages of smaller and larger quadrats for sedentary organisms. For larger quadrats, it is we can have more accurate density or distribution of organisms and better representative of the habitat. However, it is time consuming. While for smaller quadrats, we can have it easy and quick to survey the place and better representative of the organism. However, it will be less representative of organism as it is small quadrats. When ecologists try to estimate the average densities of organisms in large forested areas or aquatic habitats, they often face several challenges related to spatial scale. First, these habitats are often very large and difficult to access completely. This means that the ecologists can only sample certain areas which might not fully cover the whole habitat. Secondly, these environments are usually very diverse and uneven. Some areas might have very large number of organisms, while others may have very few or none at all. If the samples are taken from very few spots, they might not capture this variation accurately.
The number of plant species in each quadrat can be different because of several factors. Some areas get more sunlight, so they will have better soil and more water, which helps more plants to grow. Other places might be too dry, shaded, or have poor soil, so few species can survive. Human activities like walking or playing can also damage plants in certain quadrats. In some spots, one strong plant species might take over and stop others from growing. Also, not all areas get the same number of seeds from wind or animals. All these small differences affect how many plant species grow in each area.
1, 2, 3.